Hello, tonight we're going to talk about apoptosis and necrosis, both of which are a type of programmed cell death. Necrosis is the first type we're going to discuss. It's a type of cell death that occurs when the cell is deprived of oxygen or nutrients, or it gets damaged by toxins or mechanical means like a cut or frostbite. The cell dies, then the organelles lyse and the chromatin is destroyed, and then the cell ruptures. The result is swelling and inflammation, and the tissue, because it's dead, turns black. So, if you think about someone who has really bad frostbite where the tissue turns black, that's necrosis. Apoptosis is cell death that is genetically programmed. For example, sometimes it can happen when the cell or tissue is no longer needed by the organism, and so it grows it, and then the cells die. Other times, the cell could have genetic damage or could result in a cancerous cell, and so the cell goes through apoptosis to destroy itself. For an example of tissue that's never needed, when you were an embryo in your mother's tummy, you first developed, when your hands and feet developed, you developed webbing between your fingers and toes. Okay, right in here. You also developed tail. And then, after a while, those tissues go through apoptosis, and the webbing cells that were in between the fingers and toes disappear. They die off. See how it's dying in here, and then it's gone in here, and the tail disappears. All of those tissues are reabsorbed back into the body and used to grow structures that you really need. Genetic damage is a little bit trickier to deal with. For example, if the cell is exposed to some toxic substance or radiation, something like that, that causes genetic damage that is irreparable, then the cell kills itself. It goes through apoptosis. So, for example, if you go to a tanning bed and you're exposed to a lot of radiation, the epithelial cells on your skin, if they get genetic damage, they will die, and then other cells will replace them. They go through apoptosis. So, how does it all happen? How does a cell go through apoptosis? Well, first of all, in your skin, it's going to detach from all the neighboring cells. It hydrolyzes the DNA into tiny little fragments. The cell membrane forms these little lobes called blebs. And then those blebs break off into tiny fragments, and then white blood cells come and phagocytize those. They eat them. So let's watch a little video where you get to watch this happen. It's kind of cool. So let's compare the two again, just like the video showed you. When you go through necrosis, the cell is going to swell, the damaged organelles are going to swell, and the chromatin kind of get broken up and destroyed. The cell lyse, which means to rupture, and then it's going to be just spilling out into the neighboring area, causing inflammation, swelling, redness, things like that. Apoptosis, you normally have a cell called BCL2, which inhibits apoptosis. But if a signal comes from outside, that external signal inhibits the BCL, which turns it off. And because it's turned off, then the blebs form, the organelles kind of get surrounded by the blebs, and they break off, and then they're eaten by phagocytes. Okay. In plants, they have a pretty cool example of this. Your book talks about where if a plant is being invaded by a fungus or a bacteria, what the plant does is it causes the neighboring cells to go through apoptosis and die, and then the fungus or bacteria has nothing to eat because there's no living tissue to consume, and so then the fungus and bacteria die, and it helps save the plant. I think that's kind of neat. The plants are kind of smart that way. So let's watch another one where they're going to talk a little bit more about the signal transduction pathway, looking at how specifically that BCL2 is turned on and turned off. Signal transduction pathways are one of the major components that you have to understand for the AP test. So what I want you to do tonight for homework is to describe the signal transduction pathway that controls apoptosis. Okay, that's your homework tonight. Let's watch this video.
For most cells, trophic factors provide the signal blocking the path to apoptosis, programmed cell death. Trophic factor binding to its specific cell surface receptor triggers a signaling cascade, resulting in phosphorylation of the bad protein. Phosphorylated bad is unable to bind and inhibit the BCL2, BCL-XL complex at the outer mitochondrial membrane. Thus, the anti-apoptotic activity of the BCL2, BCL-XL is unhindered and the cell survives. In the absence of a trophic factor, most cells undergo apoptosis. The following is one model of how apoptosis is activated in mammalian cells. Without a trophic factor signal, the bad protein is unphosphorylated and associates with the BCL2, BCL-XL complex, inhibiting anti-apoptotic activity. When BCL2, BCL-XL is inhibited, the pro-apoptotic regulator BAX is free to permit the influx of ions through the mitochondrial membrane. The influx of ions somehow triggers the release of cytochrome C through the membrane and into the cytosol. In the cytosol, cytochrome C binds the adapter protein APAP1, which activates the caspase cascade. Caspases are specialized cysteine proteases with specific targets, such as proteins of the nuclear lamina and cytoskeleton. Cleavage of these substrates leads to the cellular events of apoptosis. First, chromatin compacts and separates, and the cytoplasm condenses. The nucleus fragments, and the cell blebs. Eventually, the cell fragments into apoptotic bodies that are phagocytosed by neighboring cells.
You may need to watch that video a couple of times to get all of the pertinent information. But again, a signal transduction pathway is important that you understand. Okay? Another thing that can control apoptosis are telomeres. Telomeres are non-coding regions found at the ends of your chromosomes. The purpose of a telomere is kind of, it tells the cell how old it is. Because every time a cell divides, the telomeres shorten. So, what exactly is a telomere? A telomere is a sequence of TTA GGG at the end of every chromosome, and you can have that 2,500 times. Okay, so it's a whole bunch of these repeated sequences. Every time the cell divides and the chromosomes replicate, some of these TTA GGGs are not replicated, and so it shortens it. Well, when it gets too short, it tells the cell, I've divided between 20 and 30 times, the cell needs to die. And so the cell dies. It goes through apoptosis and then dies. So it's just a mechanism for how the body can control how many times the cells divide, how short the telomeres get. Now, this has come into play several times. When we get to genetics, which I know is our next unit, we're going to talk about telomeres and cloning and some of the issues that have arisen. There was a sheep, the first sheep that was cloned, her name was Dolly, and she died prematurely because the telomeres, the cell that they used to create her, had shortened telomeres. It was from an adult sheep. And as a result, she died very, very young, like only 2 years old or something like that. So telomeres have a very important role to play in the lives of cells. Now, some cells in your body have an enzyme called telomerase, which is able to rebuild telomeres to their full length. The cells that have that are stem cells, and that's why scientists use stem cells to do research on, because the cells never die. They will continuously rebuild their telomeres. Also in you, bone marrow, because it divides so rapidly, bone marrow cells have telomerase. And unfortunately, unfortunately, cancer cells are the only other cell in your body that does have telomerase. So cancer cells can divide and divide and divide forever and never die, never go through apoptosis. So understanding how to kill cancer cells and how to get the cancer cells to go through apoptosis naturally is kind of an area of research right now. Well, you do have a cell in your body called natural killer cell, which is one of your types of white blood cells. The natural killer cell causes cells to go through apoptosis. It gives off a chemical signal to cells, specifically cancer cells or cells that have been infected by viruses, and tells them, hey, die already. So let's watch a little video that's going to talk about cancer and viruses.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
So external signals, like from natural killer cells, can cause signals that cause the cell to go through apoptosis. It can start those signals to go down that signal transduction pathway. Do you remember the second video we watched, that signal came to the protein, and then that caused the protein to activate the signal transduction pathway. So external signals can do it. Internal signals like irreparable DNA damage and things like that can also cause it. In either case, what happens is it causes those proteases, which are enzymes that break down protein, called capsases. It's a specific type of protease. Hydrolyze the target molecules, like those nuclear lamina, the nuclear lamina, and cause the nuclear lamina to break down. It breaks down the microfilaments inside the cell, and so then the cell breaks into those blebs and then dies. So that's the end for apoptosis and necrosis. Again, your homework is to go back through the signal transduction pathway and to describe it in detail to me. Okay? See you tomorrow. Have a great day.